Before any pharmaceutical product reaches a patient, there's one silent risk we must control: volatile impurities. They're invisible, they evaporate easily, and they often go unnoticed. Yet even tiny amounts can impact safety, stability, and compliance. In this session, we'll walk through how Headspace GC-FID helps quality assurance teams detect these hidden threats quickly and reliably, using practical strategies you can apply directly in the lab. Let's start with a simple question. Why should we care so much about volatile impurities? First, stability. Residual solvents can catalyze degradation reactions and shorten shelf life. Second, sensory impact. Even trace levels can change taste, odor, or appearance, which patients notice immediately. Third, toxicity. Some solvents carry genotoxic or carcinogenic risks, which is why ICH Q3C sets strict limits. And finally, compliance. Missing these impurities can lead to batch rejection, recalls, or regulatory warnings. So for QA teams, detecting volatiles isn't optional, it's essential risk control. So how do we actually detect these compounds? That's where Headspace GC-FID comes in. Headspace sampling means we analyze only the vapor phase above the sample, instead of the full matrix. This eliminates complicated extraction steps. Then gas chromatography separates each volatile compound. And the flame ionization detector quantifies them with excellent sensitivity and linearity. In short, it's a clean, simple, and powerful way to measure volatile impurities without messy preparation. Let's walk through the process step by step. First, preparation. We weigh the sample into a sealed vial. This keeps the matrix contained. Next, equilibration. We heat the vial, so volatile compounds move into the vapor phase and reach balance. Then injection. The autosampler withdraws only the headspace vapor, not the liquid or solid. Finally, analysis. The GC separates compounds, and the FID detects and quantifies them. Each step is simple, but together they create highly reproducible results. Now let's talk about why GC-FID is particularly suited for QA environments. Sensitivity. It can detect organic compounds at parts per billion levels. Robustness. It handles high-throughput testing with minimal downtime. And linearity. It provides accurate quantification across a wide range of concentrations. For a QA lab running routine release testing every day, reliability is just as important as sensitivity, and GC-FID delivers both. Picture a typical day in the lab. During sample preparation, you weigh accurately and add your diluent or internal standard. Then comes automation, sealing the vials and loading them into the autosampler tray. The system takes over, heating, equilibrating, and injecting automatically. Finally, reporting. You integrate the peaks and compare results against ICH limits. So instead of constant manual handling, the process becomes efficient and standardized, exactly what QA needs for consistent batch release. Even though the technique looks straightforward, method development makes all the difference. Temperature must be optimized. Too low and volatiles don't fully evaporate. Too high and degradation may occur. Time is critical. You must allow enough equilibration for true thermodynamic balance. And matrix effects matter. Excipients and formulations can change how solvents partition into the headspace. Careful optimization of these three factors ensures accurate and reproducible results every time. Let's be honest, things don't always go perfectly. Ghost peaks often mean carryover. A simple syringe bakeout usually fixes it. Low sensitivity. Check for leaks or poor septum sealing. Inconsistent results. Standardize your sample weight and equilibration time. Baseline drift. That's often column contamination or gas purity issues. Most problems are practical, not complex. Good lab habits prevent most troubleshooting. In QA, results aren't enough, proof matters. ICH Q3C requires validated methods for Class 1, 2, and 3 solvents. That means demonstrating specificity, accuracy, precision, and robustness. And during audits, documentation is key. Validation reports, calibration data, and system suitability tests must all be ready. Headspace GC-FID doesn't just help you test, it helps you defend your data with confidence. Let me leave you with one thought. Headspace GC-FID is more than an analytical technique. It's a safeguard. It protects product quality. It protects patient safety. And it protects your company's integrity. Every time you run this method, you're not just measuring solvents, you're preventing problems before they ever reach the patient. And that's what true QA excellence looks like.